начала трансляцию.
Хорошо.
Сейчас я кое-что скажу и уже знаю, как отключить звук и сделать
пусть
видео.
у вас, наверное, будет вот этот вот ОБС поверх.
Хорошо, а здесь я потом просто на это включаю, и оно
да.
автоматически все. Спасибо большое. We begin our lecture. Cytological basis of heredity. Basics of Mendelian genetics, sex-linked disorders, variation as quality of life. Plan of lecture. Futures of human genetics, Mendel's laws, inheritance of blood groups and Rh factor, the main types of interaction between allelic and non-allelic genes, multiple allelism, chromosomal theory of heredity, Morgan's theory, genetics of sex. At first, we need to speak about main terms. And first, our terms, it is heredity. It is the ability of an organism to transmit genetic information from generation to generation and implement it under certain environmental conditions. Variability is a fundamental property of living organisms to acquire new features and properties in the process to ontogenesis. Variability includes two sections: general genetics in the material basis of heredity, pattern of inheritance, organizations, futures. The next what we will speak, it is private genetics, futures of the manifestation of general theoretical laws in different types of organism. And main of our class topics, it is we will speak about medical genetics, problem and method to get result for clinic practical. A gene is a material unit of heredity, representing a section of a DNA or RNA molecule, has a specific sequence of nucleotides and carries information about the synthesis of a specific protein molecule. We will speak about two types of genes. It is dominant genes and recessive. Dominant we called a gene caused by the synthesis of a functionally significant protein and manifestation in all first-generation hybrids. For better understand, we will note this is sign as a cup, A, B, C, and so on letters. The recessive gene, it is gene which suppress it. And for understanding, we will note this is genes as a small A, B, C. So we have two types, two variant genes from mother and father. And as a result, we have two types of variant, homozygous and heterozygous, two type variants of conditions every genes. Homozygous will also two variants. It is dominant homozygous, which include both dominant genes, and recessive, which includes two recessive genes variant, and heterozygotes, which include one dominant and one recessive variant of genes. All genes which consist of all organisms we will call as a genotype, and all signs or characters of genes which we can see, for example, color of hair, color of eyes, shape of ear and other, and signs which we cannot to see, for example, speed of work enzymes, we will call as phenotype. Then we will use term as allele. This is alternative condition of dominant or recessive variant of genes. And sometimes when we will speak about one or two or three genes and describe these conditions, for example, homozygote or heterozygote, we will use term as genotypes. And we will understand that is not all genes in our organism. It is 2 or 3 or 1 genes about which we will speak. All organisms have allelic and non-allelic genes. It means if we have two variant genes which located in same chromosomes, in same locus or place in these chromosomes, so we speak about allelic genes. If one character or sign founded 2 or 3 alleles which located in different chromosome or one chromosome but different position, we will speak about non-allelic genes. The degree of expression of a gene, high, middle or low, we will call as expressive, and we use so term as penetrance, frequency of occurrence of a gene among carriers of a given gene. For our task, we will use some symbols. In this slide, we can see symbols and meaning which we use in our exercise. Mendel's laws. Mendel is a famous scientist in 19th century which made many laws and many research in classical genetics. Today we will speak about 3 Mendel's laws. The first, uniformity of first generation hybrids. This is laws about condition when crossing homozygous individuals that differ in a pair of alternative traits. All hybrids of the first generation are uniform both in phenotype, or all have a dominant trait, and in genotype, all will be heterozygotes. The second law, splitting signs. When 2 heterozygote individuals are crossed, analyze it according to one pair of alternative characters, splitting according to the phenotype in a ratio of 3 to 1 and according to the genotype 1 to 1 is observed in the offspring. Both laws, first and second, we can use only when we speak about complete dominance and when we speak about dihybrid crossing. And for this dihybrid crossing, we can use a third law. It is independent combination inheritance of traits or independent combination of genes. So, When crossing individuals differing in two or more pairs of alternative traits, independent inheritance and combination of traits is observed in the offspring if the genes that determine that they are located in different homologous chromosomes. In this slide we can see generation which will cross parents D hybrid homozygous dominant and D hybrid homozygous recessive. Between all alleles all times present interaction, and we know about two types of interaction, allelic interaction. It is maybe allelic and non-allelic genes. We can speak about allelic genes if allelic present in same locus, same chromosomes, and non-allelic genes if we speak about different place, different location on chromosomes, or different location in different chromosomes. And now we speak about allelic genes interaction. It may be some variants: complete dominance, over-dominance, codominance, gradiatility, allelic exclusion, and lethality. A type of interaction of allelic genes in which the phenotype of heterozygous does not differ from the phenotype of homozygous of the suppressive trait. This is interaction we called complete dominance. In this slide we can see two examples. When we have cup A or big A, it is dominant feature or red color of flowers, and small a, it is recessive sign or white color of flowers. And when we cross two types of flowers, white and red, all first generation will be red or will have dominance signs, dominance features. But in genotypes it will be heterozygous. It is example of complete dominance. And this example, classical Mendelian law. In this slide we can see two other types allelic interaction. For example, we will speak about incomplete, and we can see the first variant of flowers in result cross dominant red flowers which recessive homozygous recessive white flowers. We get heterozygous pink color of flowers. It is example of incomplete interaction. The next variant, it is codominance, second variant in our picture. It is a type of interaction of allelic genes in which the phenotype contains features of both dominant genes. And real, we can see in color of flowers two separate colors, white and red. The next interaction, it is overdominance or type of gene interaction when heterozygous individuals have a more pronounced manifestation of the trait compared to homozygotes. This is character excellent use in plants food. When hybrid have more and big, white and tall comparative with parents. In this slide we can see different dominant and recessive examples, complete and incomplete, which we can meet in phenotype of human. Next type of gene interaction, it is multiple allelism. It is the existence in a population of more than 2 alleles of the same type of gene. An excellent example of this type allelic interaction, it is blood group system rhesus factor. It was discovered of this phenomenon was in monkey type of rhesus and described as different types of blood. Then it was example of successful example when doctors know about different blood group in system AB0, but cannot use to transfusion blood from one people to other with one group of blood. And explain this phenomenon was in other blood system, which we now know as a rhesus factor. And now we know that more part of people in every population has antigen on erythrocytes, and part of population which not has same genes have antibody against rhesus factor antigen. People which has rhesus factor antigen we called Rh positive, and people which not have we called Rh negative. In this slide we can see that amount of Rh positive and Rh negative different in different country and different population. But in every example we can see the more part has rhesus factors. In this slide we can see that antigens rhesus factor coding by 3 types of genes. It is gene D, C and E, and all of these 3 variants genes have allelic variant, dominance and recessive. And as a result, every human, every organism has 6 allelic variant or 3 pair of allelic genes. And we will speak about rhesus factor positive when people have dominance 1, 2, 3, 4, 5 or 6 in his genotype. And in table on the slide we can see that more part of human population has dominance C and dominance D variant, little less homozygotes dominant C and dominant D, and other variants which meet in less amount. Next information, when you will know, it is about how often dominant alleles meet in human population. And in slide we can see that D dominant variant of allele we can see at about 70% of people. So often when we speak about rhesus factor genes, we can speak about D allelic of genes. The next info which we will know, it is information about rhesus conflict. What is it? It is a variant of disease when we can meet in two types. At first, it is a variant of disease when we speak about newborn and pregnancy, and the second when we can speak about transfusion of blood from one people to second. But in our slide we can see pregnant women which have three variants. The first, mother, absent rhesus factor or Rh negative, but her pregnancy with Rh positive fetus, and in first pregnancy some amount of blood or erythrocytes can present in mother organism. So organism of mother think about this type of erythrocyte as a unknown, and for save and help mother organism, immune system of mother begin make antibody against Rh positive erythrocytes. In a result, at first pregnancy we not have any effect, but in organism of mother we can find many anti-D or anti-Rh positive antigens, antibodies. Often it is happens in during process of born, and antibody more amount or high titer we can see after pregnancy. And what happens in the second and next more pregnancy? When in mother organism next fetus grows and foundation first erythrocytes with Ra positive antigens, they again can meet in mother blood, in mother organism, and ready antibody will interaction with children's or fetus erythrocytes and make their damage. So when newborns will be in our world, we can see hemolytic disease of newborns. Next slide we can see other examples of multi-allelism. It is excellent example about AB0 blood systems of blood group, but in genes of blood group we can see different allelic interactions. Here dominance, codominance, and multiple allelism. Multiple allelism, this is example. when we speak about allelic genes I0, IA and IB, 3 types of allelic. When we speak about complete dominance, we means type of genotype heterozygous. For example, if you can see a table with second blood, we can see heterozygous IA, I0, and as a result, human have second blood group of blood, and same third group of blood, IB, I0, complete dominance. And example of codominance, it is example of fourth blood group, IA, IB. Next example of our slide, it is color of rabbit. And we have 1, 2, 3, 4, 5 variant of phenotype, color of rabbit, and some variant of allelic, which give different variant of genotypes. And now we return to blood group. This term refers to the entire blood group system comprising red blood cells, antigens whose specificity is controlled by a series of genes, which can be allelic or linking very closely on the same chromosomes. First who discovered this phenomenon was Karl Landsteiner, and has been credited for the discovery of AB0 blood group system in 1900 or 1901 in different literature. And next he was awarded Nobel Prize in 1930 for this discovery. And later Jan Genestics, who was awarded Nobel Prize too, described classification of human blood groups of 4 types. In this slide we can see different of agglutinogens between 4 types of blood system. And for example, if we can see of erythrocyte type 0, we can see genes, but he not have addition galactose or acetyl galactosamine, which present in type A erythrocytes and type B erythrocytes. And when we can speak about codominance effect or erythrocytes type AB, we can see 2 variants of anti-agglutinogens or antigens, which has 2 types and not has effect suppress one other. And in next slide we can see common pictures of blood group systems. So if one organism has antigens or agglutinogens from one type of erythrocytes, and we know that all these agglutinogens can meet in our organism from salivary, from blood, from mother milk. So our organism, our immune system organism will have antibody against other variant agglutinogens which not present in our organism. So, as example, the first blood group 0. This is erythrocyte not has any one agglutinogens, no A no B. So this is organism has antibody against A, it is alpha, and against B, it is beta. This is we can see in picture. The next type blood group of second or A. We know that in this organism erythrocyte present with agglutinogens type of A. So organism will work and foundation antibody type against anti-agglutinogens B or beta. Agglutinogens B, but antibody against B called beta. And same situation in third type of blood group. We will speak about in human organism present erythrocytes with agglutinogens type B. So immune system will foundation antibody against absent agglutinogens or against A, and this antibody we will call antibody or agglutinins alpha. And in fourth blood group AB we can see both agglutinogens type A, type B, and so in this immune system not will foundation any antibody. And again we will speak about in different population and different national group we can see different meat blood group number 1, 2, 3 or 4. And in this slide we can see result of research. The total donor studied from January 2014. This is research was in 6,942 persons and around Attapurami, Andhra Pradesh. And we can see distribution of blood groups. And more often we can see 0 variant, less B, again little less A, and little amount it is type of AB. Bombay phenomenon. The Bombay or 0H phenotype is characterized by the absence of A, B and H antigens on red cells and occurs rarely, especially in tribal population in India. At first, this phenomenon was discovery of an individual devoid of H antigens on red cells in Bombay, who had antibodies in plasma reaction with all the red cells exhibition the normal red cell AB0 phenotypes. This phenotype was characterized by the absence of A, B and H antigens, and the serum of this person had anti-A, anti-B and anti-H reaction with all 0 group bloods. And these individuals were therefore genetically termed as homozygous HH recessive or Bombay phenotype. This phenomenon was reported from Mumbai, formerly Bombay, in 1952 by Bhanti. But after several other workers or researchers detect this phenomenon also in European countries. In this slide we can see classical genetics with different example of cross parents with different variant of blood group. For example, the first variant, parents 1, cross female with second blood group or AA homozygotes, and with male has third or B blood groups, but this is genotypes present heterozygotes. As a result, we have other variant blood of group in generation other compared with parents. It is will be fourth and second type, but second not homozygotes as has female. It will be variant of heterozygotes. In next variant, we cross female with first or 0 and male with fourth or AB blood group. And as a result, in generation, we can get second and third or A and B group blood systems. So, understanding of genetics of blood group systems is very important. It is very important for usually life of family and very important for transfusion. When we know mother, father and children, it will be not same blood group for type variants. But in blood systems group, not all so easy, because we know more than 33 blood group systems and over 300 antigens, and part of them we can see in this slide. It is more distribution of blood system groups, for example MNS, P, Lutheran, Kill, Lewis, Duffy, Kit, Daego, IT, Y, XG, Dombrock, and other. Sometimes it will be has a problem in transfusion when we know about complete donor and recipient blood, but has problem during transfusion, because it will be example of incomplete or incomplete in different blood group systems. Next example, it is lethality. It is alleles in which death occurs at the embryonic stage, always recessive. Dominant ones are formed as a result of mutation. They are not transmitted in a generation. And in this slide we can see the first example, when dominant yellow mutation color of mice with recessive lethal effect. And second, it is example of achondroplasia, is a dominant mutation with a recessive lethal effect in humans, caused by underdevelopment of cartilage and bone tissue. Next, we will speak about non-allelic interaction of genes. And this is example: complementarity, epistasis, polymerism and pleiotropia. But about this effect we can see in next part of our lecture.
Now we have a break time, 10 minutes, and meet in our lecture in 12:50. 12 hours, 50 minutes. Now we begin next part of our lecture.
In this part we will speak about interaction of non-allelic genes. And the first interaction, it is pleiotropy. It is the dependence of different traits on the effect of one gene. Gene W of cats, the effect of white, blue eyes, deafness, blocks the migration of melanocyte stem cells in the skin. Normally, auditory receptors arise from the same stem cells as melanocytes. One more example. In the 1936, researchers Walter Launder and Elizabeth Upham observed that chickens that expressed the dominant frizzle gene produced features that curled outward rather than lying flat against their bodies. However, this was not the only phenotypic effect of these genes. Along with producing defective features, the frizzle gene caused the fowl to have abnormal body temperature, high metabolic and blood flow rates, and great digestive capacity. Furthermore, chickens who had this allele also laid fewer eggs than their wild-type counterparts, further highlighting the pleiotropic nature of the frizzle gene. There are many examples of pleiotropic genes in humans, some of which are associated with disease. For instance, Marfan syndrome is a disorder in humans in which one gene is responsible for a constellation of symptoms, including thinning, joint hypermobility, limb elongation, limb dislocation, and increased susceptibility to heart disease. Similarly, mutations in the gene that codes for transcription factors, TBX5, cause the cardiac and limb defects of Holt-Oram syndrome, while mutation of the gene that codes for DNA damage repair protein NBS1 leads to microcephaly, immunodeficiency, and cancer predisposition in Naegeli breakage syndrome. One of the most widely cited examples of pleiotropy in humans is phenylketonuria. This disorder is caused by a deficiency of the enzyme phenylalanine hydroxylase, which is necessary to convert the essential amino acid phenylalanine to tyrosine. A defect in the single genes that codes for this enzyme therefore results in the multiple phenotypes associated with phenylketonuria, including retardation, eczema, and pigment defects that make affected individuals light-skinned. The next non-allelic gene interaction. It is a complementary gene action. A pair of genes can often work together to create a specific phenotype. We call this complementary interaction. With this type of interaction, we can see two different phenotypes instead of the four seen in two genes, one phenotype. Two or more genotype classes may display an identical phenotype. Example, two lines of poor breeding wild flowered pear plants falling into different genotypic classes. Big A, small b, cross with small a, big B, big B. This is example you can see at the slide. In next slide we can see also complementary gene action. In experience, two white parents cross and in result get same purple colors. It was one or same phenotype and different genotypes. If we will analyze it, this phenomenon, we can see both parents has dihybrid genes and have in each pair one dominant variant. The first, it was big A, at second, it was big B. In generation, in this experience was get all diheterozygotes hybrid. We need to know that gene A or allele A, it is pigment change catalyzed. Allele B from gene B, pigment change completed. So color can, if we have two dominant genes, big A and big B, when pigment change catalyzed and pigment change completed. This is variant possible only in future or generation, which we get when cross parents with only one variant of dominant genes. One more example we can see in next slide. This is example of complementary gene action, and we can see two cases, family story of disease. On letter A, we can see both parents with disease, but all their children healthy. When both parents and all their children has disease. At first, it is complementation genetic mechanism when only two or both dominant gene can addition effect and in result disease. Opposite case beta B, when dihybrid genotype parents with dominant homozygote gene A have same genotype in children. This is genetic mechanism non-complementation, and all members of this family has disease same their parents. And in the end of third part, we can make resume. Complementary, it is a type of interaction between non-allelic genes in which the trait is formed as a result of the total combination of the products of their dominant alleles. The next type of interaction of non-allelic genes, it is epistasis. Epistasis is when one pair of genes suppresses the manifestation of a trait of another pair of genes. And here we need to speak about different types of epistasis. The first, recessive, when the recessive allele of one gene masks the effect of either allele of the second gene. And the second type, dominant epistasis, when the dominant allele of one gene masks the effects of either allele of the second gene. And now we will speak about recessive epistase. And the first example, it is coat color of Labrador Retriever. And in this slide we can see at first was crossed golden and black dogs. And the first generation was all black. The next crossed was only black dogs, and in result the next generation was get 3 type of color. 9 part of black, 3 part brown, and 4 part golden color of dog. If we will speak about genotype, we can see the black color foundation both dominant allele from first and from second pair of gene. Brown color give E dominant gene, but golden color not independent from dominant or recessive gene B. Here main only recessive gene E, which can suppress dominant allele B. In this slide we can see that color of dogs codes by 2 separate genes, B, which controlled pigment production, and E, which controlled subsequent incorporation. For get black color of hair, need that both gene was function. But if gene B not function or not pigment production, not give pigment, so hair of dogs will be brown. And the second case, if mutation in gene E and this gene not function, so no pigment in coat, and it is independent present pigment or not present. So this type of hair dogs will golden. The second example is the IAB0 blood groups or Bombay phenotype. The Bombay blood group, also called HH recessive homozygote, is deficient in expressing antigen H. It means the red blood cells of homozygote recessive H blood groups has no antigen H. Often the recessive homozygote genotype of blood group is confused with the zero blood group. The difference is that the zero group has antigen H, while the homozygote recessive H gene suppress zero group. The next it is dominant epistasis, and example summer squash. At the first crossed 2 dihybrid homozygotes, one of them was both dominant genes, second had both recessive genes. At the first generation was all dihybrid heterozygote with white color. In the second cross, in generation got 3 type of color: 12 part white, 3 part yellow, and 1 part green. Look at genotype of all this phenotype. Green color was when both genes recessive. Dominant A gene give yellow color, but yellow only in case when big A combined with small b. There was 3 part from all generation. But if present big B or a dominant B gene, its gene suppressed all A gene and give color, white color, same as dominant A gene absent in genotype. In this slide we can see variation of Mendelian inheritance and how many genotype and phenotype we can meet in different gene interaction. Next, it is penetrance. Genotype does not necessarily define phenotype. The proportion of individuals with a given genotype express the phenotype determines penetrance. 100% penetrance, it means all individuals show phenotype. 50% penetrance, half the individuals show phenotype. Example, retinoblastoma, when only 75% individuals affected. And next term, expressivity, the degree or intensity with which a particular genotype is expressed in a phenotype in a given individual. And as example, we can see retinoblastoma, when some have both eyes affected, some only one. The next interaction non-allelic genes, it is modifier effects. They have a subtle secondary effect which alters the phenotypes produced by the primary genes. For example, tail length in mice. The mutant allele T causes a shortening of the tail. Not all short tails are of the same length. Another gene affects the actual length. Or in result we have variable expressivity. And the next, it is modifying environment. The environment may influence the effect of genotype on the phenotype. For example, Siamese cats, temperature-dependent color of coat. Color shows up only in extremities where the temp is lower. Enzyme for pigment formation is activity only at lower temperature. Prism, the interaction of two or more pairs non-allelic genes, the dominant allele of which affects the development of the same trait, can have a cumulative effect. And in the picture we can see example of skin color. Then more dominant genes, then more black or dark color of skin. The next, it is polymorphism. We know normal allele, it is wild type alleles. Alleles leading to pathological development of trait we know as mutant alleles from result mutations. And alleles whose frequencies in the population exceed a certain level, for example 5%, it is polymorphic alleles or polymorphism. But polymorphism of genes, it is not always pathological. It is maybe only modification, type of metabolism or characters. For example, in picture you can see the different genotype as a result polymorphism genes from gene of CYP286. And as a result we have three types of human with different metabolic activity of drugs. The combination of normal and mutant alleles determines the individual hereditary constitution. Well, Müller published a study devoted entirely to variations in size of chromosomes. Period from 1910 before 1914, Navashin demonstrated conclusively that besides a specific absolute magnitude, fixed and characteristic morphological features are inherent to every chromosome. Even in his early studies on the subject, Navashin had distinguished three types of chromosomes: U-shaped and nearly isobrachial, U-shaped and apparently non-isobrachial, and hook-like, where one arm is practically imperceptible. In 1912, Navashin established the presence of special, very fine but entirely permanent appendages connected by means to tiny filaments to the two intermediate arms of the chromosome. In 1924, Levitsky published the first of the world's guide to cytogenetics, material basis of heredity, in which he introduced the concept of karyotype. Now we know that all animals and humans have two pairs of each chromosomes, one variant from mother and second variant from father, and in result all organisms have diploid set of chromosomes. But different animals have different amount of chromosomes. This is information, short information you can see on present slide. Clutch can be disrupted during crossing over. This leads to the formation of the recombinant chromosomes. And in this phenomenon, all children have character from mother and father, from grandmother and from grandfather together. The crossover frequently is a function of the distance between genes. The greater the distance, the greater the crossover. The crossover frequency is a function of the distance between genes. The greater the distance, the greater the crossover value. The phenomenon of crossing over helped scientists establish the location of each gene on the chromosome. Grid genetic maps of chromosomes. The farther apart two genes are located on a chromosome, the more often they will diverge into different chromosomes during the farther apart two genes are located on a chromosome, the more often they will diverge into different chromosomes during crossing over. So we need more speak about crossing over. It is exchange between homologous region of non-sister chromatids. The farther the genes are located from each other on the chromosome, the greater are likelihood of crossing over. Recombinant or crossing over genes are formed. And now you can see at slide N or distance between genes equivalent sum from B, where B the number of individuals is in one crossover group, and C, where C the number of individuals in the second crossover group. All this divided by P. P is the total number of individuals, and all this multiplied for 100%. So, in result we have some percent from different distance or different size between genes, we can get different percent. But 1% crossing over is a distance of one morganida, a unit of recombination. Slide we can see different size between different genes. For example, size between A and B genes in first chromosomes only 6 morganid. Between C and B in second chromosomes it 8, and 6 between CA and AB genes consequently. In sum, it means size between C and B genes 14. In this picture, we can understand that small size between genes, it means this is genes more linked than genes which have more big distance. For example, A and M more linked than A and C or C and B. So, if size small, crossing over not will be. If size big, crossing over will be all times. In conclusion, the first, then more distance between genes, so more possible crossing over, and so linking incomplete or not present. The second, if distance small between genes, so crossing over impossible, and so these genes in full or complete linked. Next, gene clusters. It is terms about there are groups of different genes responsible for common functions and located in certain regions of chromosomes. Reflect the most important evolutionary path of organism. As example, we can see in this place about genes of immune responsible. When we have different molecular of immune system, molecules of immune system, for example, interleukin first, interleukin second, interleukin third, or tumor necrosis factors. All these molecules play important role in immune response. But all this foundation from different chromosomes. More interesting topic, it is a topic about genetic of genders. We all know about male sex has heterogametic X and Y. Full karyotype of male, it is 46 X white. In woman, the X chromosomes are represented by 2 X chromosomes that are homologous in shape and staining pattern, and normal karyotype of female 46 XX or double X. Our lecture previously we speak about body bar in one chromosomes, and we know about this nodes chromosomes which in activation. So male and female have equivalent dosage of X chromosomes. When we speak about X and Y chromosomes, not character for all organisms. For example, we know about that female homogametic, but male may be hemigametic sex, X0. So, in this case, gender will depend on the number of X chromosomes, 2 chromosomes or 1 chromosomes. This is situation, or this is example typical for worms and spiders. In medical genetics, we have situation when genes which are linked with sex chromosomes will be different from disease. For example, it may be X-linked disease and Y-linked chromosomes. Interesting that X-linked chromosomes will be interesting that X-linked or associated disease will be manifestation only from men, because they will have only one variant of allele, recessive and dominant, all times will be view, all time present. But if X-linked genes associated with disease will be dominant, so it will be variant disease which we can meet both men and women. And situation with gene associated with disease and linked with Y chromosomes will present only from men. Different example of X linked and Y linked disease you can see at the slide. We have see pedigree hemophilia.
Now we finish our lecture. If you have any question, you can ask me. And write your question in chat. If you not have a question, so for students which start to study in this week, I think you know your teacher, biology teacher, and you need join with your teacher in VK in your class group. For prepare, you will see previously lecture. Today was 5th, so we need to learn 4 previously lecture. Some of lecture we present in video, and some of lecture you will read and see PPT form. I see a question. How do scientists figure out what genes control the trait? No, usually for this use sequence. When scientists read sequence of gene, then read sequence of amino peptide, same how we made task in our class. And after sequence of peptide, we can speak about a role and which function plays this protein in our organism. Not understand question Farouk. Is that me? What means your questions? About people who joined later. I say about lecture. Farouk can use this for previously lecture for study and make it in same regime too. You need find this lecture and so or read all these materials. We can make so. When you will in your class and give this question your teacher, and teacher explain how find this all lecture and how use online resource for study. Okay? For Farouk, if this Farouk from mine group, I help in our VK group to find all lecture. Okay. Okay, if any questions not, I understand. Okay, in VK I make a connection for find all our lecture. Okay, if any question no, so I say goodbye and meet in next week in lecture and in our group. Goodbye.